Am I audible?
Yes, ma'am.
Okay, so kindly annotate your name with enrollment number. Otherwise, I won't allow you in the class. So I have to take your attendance. So first do one thing, rename with your enrollment number followed by your name, right? So I can take the attendance. Zero 01 is 03 is there. Alfia, then Tanvi Raj Parmar, you have to give your enrollment number, right?
But
Yeah.
ma'am, where we have to put?
You can see this Alfia and this, I think they have done, you can see in the participant list. Otherwise, I am not able to take the attendance. You have to just rename, right? You have to click on yourself. You have to just rename and put your enrollment number followed by your name.
Ma'am, I leave and again join. It's
Okay.
coming there.
Okay, fine. Okay, fine. And please drop the message to your colleagues as well, right? Gloria, who is Gloria? She is not able to join, I think. Let me share the screen. Okay. Are you able to see my screen?
Yes, ma'am.
Okay, fine.
Yes, ma'am.
Tanvi, still you are not able to add your enrollment number? Only 4 are there? Or students are yet to join? Do you have any idea or should I start? Okay, so I think I should start, right? I have waited for 5 minutes. So we will start with the hybridization of nucleic acid. Now, what do you mean by hybridization? Can anyone let me know? What do you mean by hybridization? Okay, so in last lecture, we have already learned about the DNA fingerprinting and what is the base composition of DNA, right? So what is the base composition in the DNA? If we talk about the nucleotide, can anyone let me know? We will first revise the last lecture and then we will move to the next lecture.
Ma'am,
Yes.
students are waiting in the meeting room. Let them in.
No, nobody is there in the waiting room, only Gloria. Only Gloria is there. See, you can see here, right? Gloria is there only. How many students are there in the waiting room? Can you let me know?
There's only Gloria, I guess, ma'am.
Yeah, I can see that thing.
And ma'am, actually, I'm traveling, so I won't be able to interact. I was going home.
Okay, who is
So
that?
I just have to.
Shaktiman. You were there last lecture?
Yes, ma'am. I'm the only guy in the.
Okay, fine. So let's start and please make sure in the next lecture you have rename. Who is this Shushant?
Yes, that is me, ma'am. That was by mistake.
Okay.
That was Shakti, ma'am.
Okay. So what is your enrollment number, Shushant?
006, ma'am.
006. So, okay. Gloria is still not here, right? I think she has some problem, I think. So only 1, 2, 3, 4, 5 are there. So let's start, right? So, okay. So what is the base composition in the DNA and RNA? Though it is online, we will definitely interact. I will not teach by one side. We need to converse, right? Otherwise, it's like just I will be speaking on and on and on, and you will be get bored. So whatever I am asking you, just you have to simply answer the question. So we can correlate with your answers to the next our topic, right? So what is the difference between the nucleotide base composition in DNA and RNA? Yes, Alfia, you want to say something?
Deoxyribose and ribonucleic acid.
Oh, that is good, but the base composition, what is the difference? I'm asking which nucleotide is present in DNA and which nucleotide is absent in DNA, or that is present in RNA and that is absent in DNA. That's what my
In
question.
DNA,
Yes,
it's thiamine, and in RNA,
uracil, right? Instead of
80C, yes.
thymine, right? Okay. So let me revise that there are 4 base pairs in the DNA. We can see 4 nucleotides A, T, G, C, and whereas in the RNA It will be replaced by uracil, right? So now in the last lecture, I have already told about the hybridization techniques. So if we talk about the DNA hybridization, it's called the southern blotting. RNA hybridization, it's called the northern blotting. And the protein hybridization, we can see the western blotting, right? So here first we will see, that is the technique, how we can hybridize using which is the molecule, right? But the molecular hybridization of nucleic acids, it involves different steps, how we can prepare the fragments and how we can hybridize, right? So one by one we will see, and what type of isotopes or enzymes we can use for the hybridization, right? What are the different types of restriction endonucleases, what we have to do, and what are the next steps. So one by one we will see, and in the lastly we will learn about the southern blotting, that is for the DNA hybridization. So the hybridization terminology, it's basically the molecular hybridization of nucleic acid. That process involves two single-stranded nucleic acid molecules with complementary base sequence. So the DNA will bind to the respective DNA which is complementary to it, right? Otherwise, it will not bind to it. So hybridization technology is a fundamental tool, especially in the, we can say, molecular biology, and has been applied in various fields. For what? Such as the cloning, if you want to clone specific cDNA, if you want to identify the gene expression, if you want to create a genomic library. Now, what do you mean by genomic library? So different fragments, DNA, different fragments can be stored as a library called a genomic library, and determining the location of gene in chromosome and diagnosis of the disease. Say, for example, I have narrated in the last lecture that different monogenic diseases are there. And if you want to identify the exact location in the chromosome of specific gene, you can always go for the hybridization. So what is the principle of nucleic acid hybridization? The technique of nucleic acid hybridization is established and developed on the basis of denaturation and renaturation. So what do you mean by denaturation? Denaturation means DNA is double stranded, right? So we have to denature. The first step is to denature the DNA and make it the single stranded, right? If you go for the PCR, if anyone has done the PCR, polymerase chain reaction using the PCR machine, right? So at 95 degrees Celsius, when we put the DNA, it will get denatured, and it will the two strands will be separate, and we will get the single strand. So our primers can bind to them, and one by one primers can aniline to the strand, and one by one DNA bases DNA pol can extend, an elongation process can be done. So first thing, that is the denaturation. Then renaturation. If you give the proper condition to the two single-stranded DNA, proper temperature, proper condition, proper environment, again it will combine, it will join back, and it will renature its own form, right? So hydrogen bonds in double-stranded nucleic acids can be disrupted by some physiochemical elements. It may be temperature. Physiochemical elements means it may be temperature, or it may be any type of element, and two strands of nucleic acid are separated into single strand. So here you can see the DNA can be from different source, right? It may be from source X and source Y. When it get combined, right, and the complementary base pair can react with each other. A will bind to T and C will bind to G, right? So automatically it will uh, bind to each other. So here you can see the first strand, that is the DNA from source X, and the second strand, right? This is the second strand. It is from source Y, but the sequence is same. Here you can see the same sequence, complementary sequence. It can be easily get attached to this complementary sequence of X. So it is a long strand. Here you can see, this is the long strand, right? But this is very short strand. It can easily find the complementary basis in the source X, and it can get bind to it. So here you can see what I mean about the denaturation. Here you can see the double-stranded DNA helix is there. And when we apply the high temperature above 95 degrees Celsius, so DNA can get denatured. And you can see the 4 different strands of the DNA, 2 from here and 2 from here. So yellow, red, blue, and green. The 4 strands are now denatured, and it will form a single strand. Now, when you give it the low temperature or the cooling temperature or the lower pH, right? Physiochemical elements or some elements, maybe you can apply the high pH, you can apply low pH, or you can apply high temperature or low temperature. So again, renaturation can be formed. Here you can see the DNA strand, again, it will be renatured in its native form, and double helix will be formed. Just a minute. If different single-stranded DNA molecules or DNA and RNA molecules or RNA molecules are mixed together in a solution and the renaturation is allowed to occur under proper condition, single-stranded DNA and RNA will bind with each other to form a local or whole molecule of double-stranded structure, as long as the single-stranded molecules are complementary. So remember the criteria number 1. What's that? The criteria number 1 is until and unless it will not get the complementary base pair, it will not bind. So the first condition is whenever it will find its complementary base pair, then only it will renature. Though you are giving the renaturation buffer, the conditions, the temperature, the pH, everything, the atmosphere, though you are providing the atmosphere, but if it will not find the complementary base pair, it will not get renatured. So along with the complementary complementary sequence, you have to provide the renaturation conditions, right? And it doesn't make like irrespective of source A, source B, source C, source D. The DNA, RNA can be from any source, but the condition is it should be complementary to each other. The specific sequence, right? Not the whole long sequence. We are asking, or we are telling about the specific sequences. Nucleic acid hybridization, that is the tool. See, it's the tool. There are different types of in the molecular biology. If we talk about the PCR, polymerase chain reactor, FISH, right? Fluorescent in situ hybridization, then agarose gel electrophoresis, then Malditos, then sequencing, then this hybridization. So these are the tools. These are the techniques by which we can analyze the certain disease, we can identify certain disease, and it should be applicable to solve the certain problems, right, for the betterment of the society. So you have to consider it as a tool or technique. So the same way, this nucleic acid hybridization, why we are telling nucleic acid hybridization? Because it may be DNA, it may be RNA, or it may be protein, right? So that's why in broad terminology, we are saying it as a nucleic acid hybridization. So this is the tool that involves the labeled nucleic acid probe. So nucleic acid probe means the same complementary sequence. If you identify the specific sequence, the complementary sequence would be your probe, but it should be labeled radioactively, or we can say enzyme labeled, right? So that is the probe. Probe is nothing else. It's not hi-fi terminology. You have to just remember whatever the sequence you want to identify, the probe you can design by your own with the complementary sequence, and it should be labeled with the some isotope, right? So here we can see it is known as the DNA or RNA fragment to bind with the target nucleic acid, which is usually a poorly understood. Still the study is going on, what is probe and how we can design, but usually yes, for the DNA chip, for the nano chip, for microarray, for mass array, all the probes we can decide. That are also the tools. Heterogeneous population of nucleic acid, a probe labeled with a detectable tracer is the prerequisite for determining a specific DNA sequence or gene in a sample or genomic DNA by nucleic acid hybridization. Yes, the probe has to be labeled. Otherwise, you cannot identify. Your probe is labeled, and when your DNA will bind to the probe, it will give certain color, it will give certain fluorescence, right? And we can identify whether our specific target is there or not. So the target nucleic acid to be analyzed are usually denatured, right? So the first step, that is the DNA, you have to take the DNA. First, you have to get denature, right? Then mixed it with a labeled probe in the hybridization system. Your probe is always labeled with the isotope or enzyme. And you have to mix it with your DNA. The probe will bind to the segment of nucleic acid with complementary sequence. जहाँ पे उसको, whenever it finds the complementary sequence, it will immediately get bind to it. The hybridization can be identified by the detection of the tracer labeling the probe. Thus, the existence of the expression of specific gene can be determined. So, if your sequence will not be there, your probe won't bind to it. And if your probe won't bind to it, then whatever the labeled system, we can say with the isotope or with the enzyme, it can't be identified. Your tracer system can't get identified. So you can get to know whether whether your gene of interest is there or not. Now, how we can prepare and label the nucleic acid? So here you have to assume that we are talking about the DNA as a nucleic acid. So how we can prepare it? So if you want to prepare your probe, right, we can easily synthesize our probe in our lab. So probe may be single stranded or double stranded, right? But the working probe must be single stranded. The stock solution, it can be the double stranded, but whenever the working probe, it must be single stranded. Why it is single stranded? You need to made it complementary to your, and you have to get exposed that site, so your gene of interest can bind to it. So it must be single stranded. The probes used in the hybridization of nucleic acid includes a very small fragment of the DNA. We can say only 15 to 50 nucleotides. It should not be small, it should not be very larger, right? So the range between 15 to 50 nucleotides you can synthesize in your lab, and that is the very, we can say, average genomic DNA fragment. It can be cDNA fragment or it can be RNA. So probe you can design using the three different nucleic acid. It may be DNA, it may be cDNA, it may be RNA. Now, what is cDNA? Can anyone let me know, please? What is cDNA? Nobody knows? Okay. So whenever this is the COVID era, right? So COVID has the genetic material what? mRNA, right? So if you want to get it convert into the cDNA, so what you have to do? There is a reverse transcriptase enzyme is there. You can always put the reaction and mRNA get converted into the cDNA. It has to just make the complementary copy, right? So whenever you want to amplify the viral particle, first you have to isolate the mRNA, you have to make it into the cDNA, and then you have to put it into the RT-PCR. Then you can say the viral load, that viral load is 9, 10, 12, 15 or 21, right? So cDNA is just complementary DNA of mRNA, right? So it is the using the reverse transcriptase enzyme, we can convert mRNA, any mRNA to cDNA, and we can see mRNA is very unstable. You cannot store mRNA as it is. Its stability is very low. So immediately, whenever you isolate the mRNA, you have to convert into cDNA, and then you can put into your -20 refrigerator, right? So yes, so you can design your probes using these three nucleic acids. It may be DNA, it may be cDNA, or it may be RNA. But the condition is it should not be longer. It should not be very less 10 nucleotides or 15 nucleotides. You have to keep minimum around 25 to 50 nucleotides, right? 15 is also too low, I think. 22, 23 nucleotides generally we can keep. So this is this is the theory part, but in research, what we have done till date, right? So I am sharing my knowledge with you. In our lab, we used to make a probe of 22, 25 nucleotides, right? Oligonucleotide probes are short, we can see single-stranded DNA fragments designed with a specific sequence complementary to the given region of the target DNA. Say, for example, if you want to identify a specific segment of the gene, any gene, say, for example, some part of the VEGF gene, right? So you have to take the sequence of that gene, and how you can synthesize the probe? The same sequence, you have to take the complementary sequence of your gene, and you can synthesize the probe, right? So that is, it should be complementary to your desired, your interest of gene. They are usually synthesized in vitro. Okay, so the terminology you must know about the in vitro and in vivo. In vitro means in the lab, right? Whatever we are doing in the lab, in the cells, artificially, right? In our body, whatever we are, our body system or our cells, they are designing or they are making or they are synthesizing, it's called the in vivo, right? In mice, it's the same experiment. If I would like to apply in the mice, it's called the in vivo experiment. But the same experiment, if I would like to continue in the lab, in the laminar air flow, in our lab, it's called the in vitro, right? So in the lab, we can say in vitro, or in the human body, in the mice body, in the system, we can say the in vivo. So here in vitro, we can synthesize the probe. Now, there are three different types of probes: genomic DNA probes, cDNA probe, and RNA probes. So genomic DNA probes can be prepared from the cloned DNA fragment in plasmid. We all know about the plasmid, right? What is the plasmid? It's a double circular DNA present in the prokaryotes, in the especially bacteria, right? And it is used for the cloning purposes. If you want to go for the recombinant protein, if you want to produce the insulin protein, if you want to produce the interferon protein, so you will always clone that gene into the plasmid, and that plasmid will that DNA will be converted into mRNA, and we will get the protein insulin, and it will be sold in the it will be stored in the inclusion bodies, and that inclusion bodies we can solubilize, and we can get the insulin back, right? So here we can synthesize the DNA probe in the plasmid, which is cloned, right? The cDNA probe we can prepare from the cloned cDNA in plasmid. The same thing. If you can clone the DNA in the plasmid, you can clone the cDNA as well. This using the same procedure, we can make a cDNA probe, and then afterwards, how we can get the cDNA? The same. I told you, using the mRNA, we can get the cDNA. And RNA probe are usually transcribed in vitro from a cloned cDNA in a proper vector. The size of genomic DNA probes, cDNA probes, and RNA probes may be 100 base pair to 1000 base pair, right? Labeling of probes. Now you have synthesized the probe. You have selected your gene of interest, gene of target in the laboratory. Say, for example, you have designed your DNA probe, right? Now the probe is ready, but how you will label the probe? So it is usually labeled with a detectable tracer. It can be detected it can be detectable, right? Either isotopic or non-isotopics. It may be radioactive or it may not be. The purified oligonucleotide is labeled in vitro by using a suitable enzyme, or it may be labeled with the suitable enzyme to add the labeled nucleotide to the end of the oligonucleotide. So your oligonucleotide will be labeled using different types of radioactively labeled molecule. In the next slide, we will see how we can get the labeling. So labeled RNA probes are usually synthesized by RNA polymerase, right? So RNA polymerase in the presence of ATP, GTP, CTP, and the labeled UTP with specific fragment of the gene or cDNA in a proper vector as a template. So RNA probes can then be generated and be labeled at the same time. So the same story will goes with the DNA, with RNA, and with cDNA. How we can label the probe? If we talk about the genomic DNA probe and cDNA probe, usually labeled in the process of DNA synthesis in vitro. Whenever you are synthesizing the probe, at the same time, you have to get it labeled. In the reaction of DNA synthesis with a DNA probe as template, if a labeled DNTP, which can be incorporated into newly synthesized DNA chain, it is added as a substrate and the labeled DNA probe will be formed. So whenever you synthesize the DNA, see, this is the 5' to 3' and 3' to 5'. Whenever DNA polymerase will come and will extend the nucleotides, right? So instead of GTP, instead of CTP, we have to incorporate the labeled GTP, labeled CTP, right? So nucleotide will be labeled, it will be incorporated into the newly synthesized strain. So your probe will be fully labeled. Got it? Some sensitive detecting methods for each of the labels used in the nucleic acid hybridization are there. There are different types of methods, so we can validate whether the probe we have synthesized it is correct or not, right? Sometimes it may happen that instead of labeled DNTPs, unlabeled DNTPs can be incorporated, but you have to make sure that every time when you synthesize the probe, the DNTPs must be labeled. So after hybridization, the location and the quantity of the hybrid molecules can be determined. The labels in common use include radioactive. So with the DNTP, which labels we are using, right? So P32 and S35. Mostly radioactive phosphate and sulfur, P32 and S35, we will use as a label for the DNA probe, and non-radioactive, some biotin and fluorescent substances which are used as the label DNTP. Now, what happened when we actually hybridize the nucleic acid? Now you have your gene of interest and you have your probe ready with labeled isotope, right? So both the things now you have. Now how you can hybridize the nucleic acid? So if we talk about the southern blot hybridization, especially for the DNA hybridization. So the main application of the DNA hybridization is you can identify the gene expression, your desired gene of interest. You can identify the disease, right? Specific monogenic disease. So southern blot hybridization is the assay for sample DNA by DNA hybridization, where the two complementary strands of DNA can be hybridized. It detects target DNA fragments that have been size fractioned by gel electrophoresis. Now figure 4.141, if we say, what is that? Okay. Here you can see this, all the written portion I will explain you here, right? So you don't have to get read. So first you have to take the target DNA over here, right? Now, already I have explained you in the last lecture that you have to digest your DNA with the restriction endonucleases, right? So you will get the random cut, right? And you will get the specific pattern of your digested DNA. Now that mixture of the fragmented DNA, you have to load into the well of the agarose gel electrophoresis. So here you can load your sample. DNA will migrate. DNA will migrate on which basis? The lighter band, we can say, the very small size of DNA will move faster. When we compare with the large size of DNA band, it will move slower, right? Because molecular weight is high, so it will move slower. And the molecular weight of the DNA, if is very low, then it will move the faster, right? So based on the size, we can see if say for example, if I'll give you the example of 100 base pair and 500 base pair, which DNA molecule or which band will move faster? Gloria, if we have loaded two different types of DNA which has the size 100 base pair and 500 base pair, so which fragment will move faster? Gloria, are you there?
Yes, ma'am.
Okay, so can you give me the answer?
I'm not sure, ma'am.
Why? So whenever you are not sure, you have to ask me. You can interrupt me in between that, madam, we are not getting. So I will explain you again and again, right? So there are two different types of DNA, right? If the molecular weight and the size is different. So the larger size will move slower and the smaller size would move faster. So the smaller size, that is the 100 base pair, and it will move faster, right? So you will get the uh, specific uniform pattern. And here, whatever the band pattern you will get, you have to denature that in alkali, that is in NOH, strong alkali. The same gel you can apply and you can transfer onto the nitrocellulose or membrane, nylon membrane, right? Whether you can use the nitrocellulose membrane or nylon membrane. So your DNA will be transferred over here. The same thing. Here you can see the probe. You have synthesized the probe. You have to radioactively labeled probe. Now you have this DNA sequence labeled with the radioactive. So it is radioactively labeled probe. Again, this probe will be denatured, so you will get the single-stranded probe. Now you can hybridize. Here you will get the probe that is radioactively labeled, and here your DNA on nitrocellulose membrane. So when they both will be, we can apply the same thing over here. You can get the hybridization. The excess or unhybridization, excess or we can say unhybridized molecules will be washed off here. And when you apply the X-ray film, you will get this hybridization pattern, right? So final, you are the things what you want, actually your desired interest of gene, your target, your gene of interest, or you will get over here. This is your hybridization result, right? So this figure 4.1, it narrates this slides, these 4 slides, right? So if we talk about the southern blot hybridization, the target DNA is first digest with the restrict endonuclease. Again, I am repeating, whosoever are not getting, please let me know. Because see, it's an online mode, and in the offline, we used to write with the chalk and board and we can explain you. But here you have to tell me that, madam, please repeat, we are not getting. So I will definitely repeat it, right? So, okay, so let me go step by step. First, what we will do in the sudden blotting, we will isolate the DNA first. We will use the restriction endonucleases, right? So we will get the fragmented DNA. Now, this fragmented DNA, we will load with the agarose gel electrophoresis. And the same gel we will transfer with the nitrocellulose membrane or nylon membrane. Now, these DNA fragments are negatively charged because the phosphate group so to migrate towards the positive charge. So the shorter DNA fragments move faster than longer ones, and the fragment between 0.1 and 20 kb in length, the migration speed depends on the length of the fragment. So remember this thing, that always your migration pattern depends on the length of the DNA. Now, after the electrophoresis, the gel or the DNA fragments are denatured with a strong alkali, right? And these fragments will transfer to the nylon membrane. It will be immobilized onto the nylon membrane. It will be stick to the nylon membrane, right? Then it will be react with your designed probe, which is radioactively labeled. So the probe will bind only to the complementary DNA sequence. Now you have the nylon membrane, you have the probe. Now when it will, we can see we put the hybridization buffer and all the reagents, it will bind to only the complementary sequences. As the position of the immobilized single strand target DNA fragments on the membrane are faithful records of the sieve separation achieved by the agarose electrophoresis. So now they can be related back to the original gel to estimate their size. Again, we can estimate the size using the same thing. So here, whenever now the things are getting hybridized, right? Now you have to expose the X-ray film and you can see the hybridization pattern, right? So this is the figure number 4.1. Okay. So we will, okay, how many? I think 23. Okay. So till here, if anybody need to understand and make it clear, if you don't get yet, please let me know. I can repeat it. But it should not be like that, madam, we are not getting, and I don't want to carry forward the same situation again and again. Hello? Everybody get it? What is?
Yes, ma'am.
Okay, fine. Okay. So next, southern blot hybridization is widely applied in researchers since its invention. Routinely, we are using this southern blot hybridization to identify the specific gene, right? Gene of expression, screening of recombinant proteins, plasmids, mutations, specific DNA, identification of the specific DNA, or identification specific pathogenic organisms. And if we want to go for the restriction mapping, right? So the basis in DNA only pair with a specific G. We all know that A pair with T and G pair with C. And in the short DNA sequences, yes, the short DNA sequences will not be entertained and the large DNA sequence will not be entertained, right? Because whenever you design the short probe or short DNA sequences, it can bind with many places. And the with the large DNA sequence, sometimes it will be misleaded, right? So moderate DNA sequence, as I told, 22, 25, 30 nucleotides will be optimum. The source of any single DNA of DNA is irrelevant. Merely the sequence is important. Means you can hybridize any source of DNA, source from X, Y, Z, whatever the source, it is irrespective, right? You just make sure that your gene of interest and your probe should be the complementary to each other. This phenomenon of base pairing of single-stranded DNA strands to form a double helix is called a hybridization, as it may be used to make hybrid DNA composed of strands from different sources. Because DNA sequences will seek out and hybridize with other sequences with which they base pair in a specific way. Much information can be gained about unknown DNA using single-stranded DNA of known sequences. You have known sequences and now you can target the unknown sequences, and this hybridization we can get to know the specific disease, specific pathogenicity of the organism. Short sequences of single-stranded DNA can be used as probes to detect the presence of their complementary sequence and number of applications. So these probes can be used in the southern blot, right? For northern blot, for in situ hybridization, for dot blot, In the addition, the renaturation or hybridization of DNA in solution can tell much about the nature of organism of the genome. Now, what is library screening? Library means what? The most common method of library screening involves hybridization of probes to target DNA. So whenever you make the library, you have to screen, you have to go for the first hybridization process, and it refers to the specific way DNA sequence may pair with the exact complementary sequence. Now we all know about the probe. Probe is radioactively labeled. You can synthesize probe in vitro. And for the designing of probe hybridization screening, something must be known in advance about the target sequence. Until and unless you don't know your target sequence, you are not able to design your probe. So the first requirement of your probe designing is you should know the target sequence. What you would to identify, how you would identify, and why you want to identify. So you have to make clear your goal, your target. If you want to know the certain disease is present or not, so the gene for that specific disease, the sequence should be known, then only you can design that probe, right? And this screening, hybridization screening, takes advantage of the fact that complementary strand of DNA can recognize one another by sticking DNA from many colonies or plasmid in a library to membrane. Any type of colonies you can, it will stick and it will hybridize. The DNA will be single stranded, hybridizing the probe to the DNA on the membrane. The same method follows, right? The colony, the single stranded, we can see the hybridization process. Here you can see the hybridization screening of the colony, right? So first you have to take a membrane, whatever the cells, these are the cells, your colonies, right? So this is your target clone. Here you can see the target clone is here. So you have to make a probe using this target clone, right? So you have to synthesize your probe using this target clone. Now the all cells will be transferred to the membrane, cell will be lysed, shell will be washed, right? Then you have to transfer to the membrane, and that membrane will be blocked for the some unwanted hybridization, right? Now you can add the probe. So whatever your desired clone, that only it will bind. Here you can see only your colony, desired colony will only bind because you have already synthesized the probe against this colony. So whenever you apply the X-ray film, you can, this is your colony, desired colony is there. The same way. First, in nutshell, if we revise the southern blots, first you have to isolate the DNA, digestion of DNA with restriction enzymes, separate the DNA fragments by gel electrophoresis, transfer the fragment to the nylon paper, hybridization with the probe, and probe detection. This is the auto red development when you expose to the X-ray, right? So making a southern blot 1 and 2 digestion, here you can see, this is the marker. This is the fragmented version, right? Fragmented means DNA will be, your DNA fragments will be generated using restriction endonuclease. But this is the marker, this is the control, and this is the experiment. Here you can see this is the marker, this is the control, and this is the experimental. Now making a southern blot 3. This is the marker, control, and experiment. Now this DNA will be transferred to the membrane, right? So this, all this buffer and all nylon paper, and you can put all this reagents together, it will be transferred. In blot 4, you have to wash, you have to block membrane with bound DNA. Now this is the membrane with bound DNA. All the DNA will be transferred into the membrane. Then parts of the membrane not already covered with DNA now bind blocking reagent. Whenever you add the blocking reagent, means wherever the DNA will not bind, it will block with the blocking reagent, right? Now probe, when you add the probe and DNA will be bind together with the complementary. And here you can see this is the probe after washing. When you apply the X-ray film, this is your desired, this is control, and this is, you can see your experimental, right? Complementary DNA. So here you get your experimental DNA, right? So membrane with probe bound to complementary DNA. Now X-ray film is exposed and fragments complementary to the probe appear as a band on the auto rate. So here we can see here your hybridization will be take place. Control, and this is the experiment, right? So this is all about the we can see southern blotting, DNA hybridization, right? So here it is the marker. So we can get to know, okay, what is the size of this? It may be 100, it may be fund. If it is 500, so around 550, we can see 550 base pair is there, right? So by using this marker, you can identify the size of the your desired fragment. Okay. Now I think this southern blotting is over, but I need to take the attendance. Wait. How many of you are there? I think stop share. Yes. Okay, only 5 are there. 1, 3, 5, 9, 10. 06. Who is 06? 1, 3, 5, 9, 10. Gloria? No, 006. Who is that?
Sushant.
Sushant. Okay. 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 So if you have any question, then in the next lecture, please come with your questions. So I'll try to solve, right? But yes, you have to read it once again, right? You can read research paper, you can read PPTs, you can read your books. But yes, whatever the doubts you have, anytime you can message me. I'll try to solve this, right? Okay, then. Have a nice time. Bye.
Thank you, ma'am.
Thank you, ma'am.
Okay, bye.